I am going to start by just mentioning quickly, I wanted to thank Tanya for the opportunity to speak to the ERCC. Our paths crossed this past summer at a keystone meeting dedicated to understanding the biology and the function of extracellular vesicles. And I think the take-home message that I took from that meeting is that this field has literally exploded in just the past few years. So the biology and the interest in the area is just growing. There's implications in cancer biology, neurodegeneration, as well as a number of normal physiological processes as well seem to be regulated by this unique form of intercellular communication. And today I want to sort of focus on the work that we've been doing in one area of this in the cancer biology realm. Also, I was told to mention just that my email address is down here in the corner in case there are questions that come up that people would like to email me. I'll be sure to get back to you in correspondence. So with that, I'd like to get started by giving you a little bit of background into the interests of the laboratory. And what I have been studying for about 20 years now is understanding the fundamental mechanisms that drive the growth and survival of cells, and in particular, understanding how those processes relate to cancer development as depicted here on the left. Now, one of the critical mechanisms that drives cell growth and survival involves cell-to-cell communication. And the classical example of this is soluble or secreted diffusible factors that are generated by cancer cells. So those come in a couple of varieties. They could be growth factors. They can also be pro-inflammatory cytokines. And they can communicate with neighboring cells. And as depicted down here is a sort of a diagram of a typical tumor microenvironment of any kind of solid tumor. And in it, you'll see there are tumor cells that are next to other tumor cells, as well as normal cells that are known to reside within these tumor microenvironments. And what happens is that these secreted factors, in these cases, these growth factors, they can travel a certain distance after the tumor cell has secreted them. And then if they come in contact, for example, with another tumor cell, they can start signaling events in these tumor cells that cause them to grow and divide. And certainly you can understand how a tumor mass could be expanded by increasing the proliferation rates of the cancer cells. But equally as important are the effects that these soluble factors tend to have on normal cells. And as pictured here is an endothelial cell. So once again, many of these same growth factors can bind to their corresponding receptors expressed on the surface of endothelial cells. This causes the endothelial cells to migrate towards the tumor and promote blood vessel formation. And it's known now that tumor angiogenesis, of course, is an important way that to sustain tumor growth. And so this is always for about 40 or 50 years now has sort of been the classic ways that cells communicate with their environment. But as I had started, excuse me, talking about a little earlier, the appreciation that cells are also forming and shedding what are referred to collectively as extracellular vesicles is now becoming appreciated as another major form of intracellular communication. And so in this top diagram here, you have on the left a donor cell and on the right a recipient cell. And you can see that there are, as depicted, these lipid-enclosed vesicles that are being released from the donor cell. And they come in two different classes. They're what are referred to as microvesicles and exosomes. And there are a couple of ways that these two classes of extracellular vesicles are different. The first of them is in how they're formed, or their biogenesis. So microvesicles are formed as a result of a direct budding up of the plasma membrane. They're then loaded with specific cargo and then released into the extracellular space. On the other hand, exosomes are actually formed as a result of rerouting of the typical endocytic processes that occur in all cells. So what happens is small fractions of the plasma membrane are usually invaginated and break off into endosomes. Those endosomes typically accumulate into multivesicular bodies, or MVBs, as depicted here. And typically those MVBs are directed to the lysosome for degradation. However, after receiving certain signals, these MVBs in some cases are rerouted to the cell surface. they fuse with the plasma membrane, and these endosomes are released into the extracellular environment. And at this point, they're referred to as exosomes. So one of the common features, though, between these two classes of extracellular vesicles, including microvesicles and exosomes, is their cargo. And it's also what makes them rather interesting. So it turns out that in addition to just containing certain growth factors and extracellular matrix proteins, they also contain a number of other proteins and molecular signatures not thought to be secreted by viable cells. And these include a number of cell surface receptor tyrosine kinases, cytosolic signaling proteins, numerous nuclear proteins, including transcription factors, and then there's a whole host of RNA species that also find their way into these extracellular vesicles. And that includes RNA transcripts, microRNAs, and even long non-coding RNAs appear there. Now, we have focused primarily on the proteins that get involved, but again, there's a lot of work that's been done to study the RNA and their roles in these extracellular vesicles. So what happens is these vesicles, once released into the extracellular space, they can dock onto nearby cells, and when they dock, they can initiate some signaling events that are important for causing phenotypic changes in recipient cells. Also, what happens is the cargo within the vesicle gets transferred into these recipient cells, and sometimes that's also responsible for the phenotypic changes that are associated with these extracellular vesicles. Now, the other important difference that I wanted to mention between these two different classes of extracellular vesicles, microvesicles and exosomes, is their size. So microvesicles tend to be much, much larger than exosomes. So microvesicles average between 0.2 and 1 to 2 microns in diameter, whereas exosomes are between 30 and 100 nanometers. And just to give you an idea of what microvesicles budding from the surface of a cell looks like, this is a picture over on the left-hand side of a scanning electron microscopy image of a highly aggressive breast cancer cell line known as MDA-MB231 cells. And you can see that this cell is literally covered in microvesicles that are budding off the surface of this cell. Now, I'm going to be using this cell line quite often throughout my talk, so I'm just going to refer to it as 231 cells from here on out. So I had mentioned to you about the differences in the sizes, and our lab has taken advantage of those size differences to purify microvesicles and exosomes and separate them out from different biological solutions. And in particular, we do a lot of work with conditioned media off of cancer cells. So, excuse me, how this proceeds is you take the media, conditioned media off of any plate of cells you want to study. We go through a series of low-speed centrifugations, and this removes cells and debris. And at this point, if you look at the vesicles that are present in this conditioned media using dynamic light scattering, we can see that there's two distinct populations. There's one that averages about 30 nanometers, and a second one that averages about 400 nanometers in diameter. And then what we do is we pour our conditioned media over a 0.2 micron filter. So what this is going to do is it is in effect going to split these two populations of extracellular vesicles into two populations. The larger ones, this 400 nanometer peak, we're going to remain on the top, and that's what we refer to as our microvesicle preparation. Whereas the smaller ones are the exosomes, and they're going to actually flow through the filter. And you'll see in the second part of my talk that we actually do one additional step to isolate the exosomes from the conditioned media. So in the first part of the talk, we're just going to focus on the microvesicles. You can see we have a nice clean preparation here by dynamic light scattering after we put them on the filter. And then we can use those purified vesicles for Western blotting or biological assays. And an example of that, of putting this approach to use, is shown down here in B and C. So over in B, we have, again, our aggressive breast cancer cell line, these 231 cells. This time we've expressed a plasma membrane targeted GFP construct in them. So you can see that the membranes of these cells are glowing, are fluorescent green. We can see vesicles budding up all over the surface of this movie. And with this arrow, you can actually watch one of these fluorescent vesicles actually breaking off the surface of the cell. If we then take the conditioned media off of these transfected cells and we isolate the microvesicles, we can detect the plasma membrane targeted GFP construct in them. also on blots and on every single one of our blots, we do two important controls. We have a marker that's for extracellular vesicles called flotilin-2, so we can detect flotilin in each of our preps. That's confirming that we have isolated extracellular vesicles. And then we also always probe for a protein that isn't incorporated into these into microvesicles or exosomes, as you'll see later in the talk. And in this case, it's I-kappa B alpha. This is a signaling protein in the NF-kappa B pathway. It's expressed in these 231 cells, but it isn't found in the microvesicles. And we use that to show that our preparations are devoid of any cytosol contamination. And so you'll see that on each of our blots, but I won't talk about it too much beyond this point. So what do these vesicles do biologically? Well, we have shown, and others as well, that if you take the microvesicles that are shed by a highly aggressive breast cancer cell, like 231 cells, and then you add them to recipient cancer cells, they can promote their growth and survival. But what we became interested in what happens if you do the same experiment, but in terms of the recipient cell, instead of being a cancer cell, we use a normal cell type that might be typically found within the microenvironment of a breast cancer cell. And in this case, we asked, how would these microvesicles affect fibroblasts, one of the major cell types that are non-cancerous cell types that are found in the tumor microenvironment of breast cancer cells. And we've done a number of experiments on them, and they change in a number of ways, but the one that I just wanted to highlight here is shown in B. And this is a soft auger colony formation assay, also known as anchorage-independent growth assays. And what's important to know about these is that normal cells, like a non-transformed fibroblast like NH3T3 cells, will not grow in soft auger. So they remain as these small single cells all throughout this matrix. However, if we take this same cell line and now we put them in soft auger and we incubate them with microvesicles derived from these aggressive 231 cells, now these cells start to grow and form colonies in soft auger. So this was really surprising to us because it really starts to argue that these vesicles have this incredible capability of conferring onto normal cells the transformed properties of a cancer cell. And we have gone on and then to identify the key, at least some of the key players in these aggressive breast cancer cells that are responsible for mediating this effect. And it includes a cross-linking enzyme that is known as tissue transglutaminase, or TG, and also the extracellular matrix protein fibronectin. So as depicted down here in A, you can see that these cancer cells are shedding these microvesicles, that there's fibronectin and tissue transglutaminase associated with their membranes. And what we have found is that this cross-linking enzyme actually cross-links fibronectin into an oligomer, and that allows these microvesicles to dock onto normal recipient cells and start aberrantly signaling, aberrant signaling pathways in these normal recipient cells that causes them to transform. And so certainly you can start to appreciate how microvesicles can be impacting the microenvironment by causing non-transformed cells in the local environment to start to behave like cancer cells. Now, that work we published now 6 years ago, or 5 years ago now, and since then we've also identified a key signaling pathway which are important for making microvesicles at least in aggressive cancer cells, and it involves the small GTPase Rho A signaling through to ROCK kinase to additional proteins that cause cytoskeletal rearrangements that cause the budding up of the plasma membrane that's associated with microvesicles in the shedding. And so we've gone on, of course, and then of course this is mechanisms that we can manipulate in cancer cells. And this work, combined with the work of a number of other people, have really shown how important these extracellular vesicles can be in terms of promoting cancer progression. So in addition to changing some of the normal cells in the local environment to behave more like cancer cells, the vesicles have also been shown to promote tumor angiogenesis. They've been shown to promote cancer cell migration and invasion, as well as promoting metastasis. So really, every aspect of cancer progression is in some way linked to these extracellular vesicles. So in addition to sort of the understanding the biology of these extracellular vesicles and how they're made, there is another important part of the research that's going on in the extracellular vesicle community, and that's their use for diagnostic purposes. And a lot of this interest is really stemmed from the discovery that cancer cells can shed extracellular vesicles in the circulation of cancer patients. And this is quite surprising. People don't really appreciate, they might be involved in metastasis, but how they get there initially isn't fully understood. But a number of large pharmaceutical companies, as well as startup companies, as well as clinical researchers, are all really chasing these things to try to see if they could be used for diagnostic purposes. And there are a lot of studies out there identifying potential biomarkers that could be used to identify patients with a particular kind of cancer or a certain grade of cancer. But one of the things that's lacking in the field is a early and general marker that might be present in these EVs, and can we use them to detect cancer in patients very early on. And that would be critical because it's well appreciated that if you diagnose cancer early, you have a better chance of treating it and patient survival, and also general. So I had mentioned that a lot of these biomarkers are specific for certain types of cancer, or even in a subset of cancer, of certain type of cancer. And what we need is markers that could be found in all vesicles that are generated by cancer cells. And that is where our sort of basic biology approaches and understanding how the vesicles are working are going to be coupled now with this idea of how do you best use these extracellular vesicles for diagnostic purposes. And so the question that we come up is how do EVs generated by cancer cells differ from those generated by normal cells? And there's been basically two major ways that have been proposed of how this can occur. The first is shown here on the left is EV amounts. So it's been reported that cancer cells generate large numbers of extracellular vesicles, whereas their normal cellular counterparts are able to produce vesicles, but at a much lesser degree. So one of the ideas is that if you have sort of a threshold that's normally occurring in the blood, and that number of EVs in the bloodstream from a patient is increased beyond a certain level, that maybe that could be used as an indicator of cancer. Now, the second way that EVs might be used is by looking for distinct cargo that is expressed only in the extracellular vesicles derived from cancer cells. So I'd already mentioned that normal cells make extracellular vesicles. They are in the bloodstream circulating of healthy individuals. But the cancer cells are also making their extracellular vesicles that make their way into the bloodstream. And what we're looking for here is just depicted by this red rectangle, is something that's unique for them. And so in either case, if we could find how cancer cells are different from these normal cells, that could give us an indication of how, of diagnosing cancer early on. And so we set out to sort of start to try to answer these questions using an inducible model of cellular transformation. And for this purpose, we took advantage of a long history in the lab of working on the Rho family of small GTPases. They function as molecular switches in human cancer. So they go between an inactive GTP-bound state and an active GTP-bound state. And when they're hyperactivated, they can promote the transformed phenotype. Now, what's interesting about these is there's a number of regulators that influence the activation of Rho family members. And one in particular, known as DIBL for diffuse B-cell lymphoma, is known as a GEF, or guanine nucleotide exchange factor, or an activator of the Rho family of small GTPases. And if you take basically the full-length version of this protein, which is referred to over here as proto-DIBL, you can see that there's a large red region. This is a regulatory region, and it's followed by a DH and PH domain. This is a DIBL homology, plextrin homology domain. And it's this DH-PH domain that's responsible for converting inactive small GTPase to the activated form. And this large red region inhibits this process and it's tightly controlled. So if we express this full-length protein in normal fibroblasts, and this is a focus formation assay, you can see that it doesn't cause flow cytoform, so it's a non-transforming protein. However, if we express a construct of Dibble, which we refer to as OncoDibble, that's lacking this large regulatory region, now it just has the DH and pH domain, it's constitutively active, can activate the Rho family of GTPases and cause them to transform, as seen here in this lower right-hand column, the plate labeled OncoDibble. You can see the foci building up. So we took advantage of the fact that the OncoDibble, being such a strong transforming protein when expressed in cells, is what we decided to use as a model system. And so what a senior researcher and associate in the laboratory did, John Erickson, is that he made an isogenic model of cellular transformation where he introduced a plasmid that is regulated, is controlled by the presence of doxycycline into meths. And then and you can see that as long as these cells expressing this construct is in the presence of doxycycline, they don't express this HA tagged form of OncoDibble here. However, if you just remove doxycycline from these cells, you can induce expression of OncoDibble. And in fact, you can transform the cell. And two ways that we determine that is we perform soft auger analysis on these meths that were either induced to, either not expressing so they're normal or induced to express OncoDibble. You can see that only the OncoDibble expressing meths form these large colonies in soft auger, and the uninduced meths do not. It's quantified here. And then we also performed a survival assay. So if you take normal fibroblasts and you expose them to serum-starved conditions, they'll undergo apoptosis, as shown here. However, if we first induce OncoDibble expression in these meths, now we can suppress this apoptotic response. So these are what we would refer to as highly transformed cells. Now we wanted to take this model, and going back to our initial question is, how might the extracellular vesicles that are generated by cancer cells differ from those generated by normal cells? And we decided that what we would do is we would induce OncoDibble expression for a very short period of time. So just as these cells are going from basically a non-transformed state to a transformed state, is when we wanted to look at the vesicles that these cells were generated and see how they could be different. So this would be a very early stage event in cancer progression. So the first thing we did is we took both of these cell types and we labeled them with a fluorescent membrane dye. And you can see in these inset boxes that both of these cells are generating vesicles that you can detect all along their surfaces. We then isolated the vesicles, microvesicles, from the conditioned media that are shed by these cells using the filter method that I had mentioned to you earlier. And we can again look at by Western blot, and you can see flotilin is present in both. So both of these cell types are capable of generating extracellular vesicles. The sizes of these vesicles is determined by EM. We're about 400 nanometers in diameter on average. And then the really interesting thing we found is we wanted to quantitate the number of extracellular vesicles that were produced by each of these cells. And as you might remember earlier, one of the differences that has been reported is that cancer cells should be making a lot more than normal cells. But we had found in our model system, you could see here in blue is the uninduced cells, and in red is the OncoDibble expressing meths. That what happens is that they make roughly equivalent amounts. If anything, the uninduced cells seem to make more microvesicles than the OncoDibble expressing cells. And so this was really one of the first indications that we had that maybe vesicle numbers is not a reliable readout of cancer, especially probably at the very earliest stages, that vesicle numbers don't necessarily change. Maybe in higher grade or more aggressive cancer cells that could be, but it probably is not going to be a reliable readout, at least in all cases. So the next thing that we wanted to do is we wanted to look at how these extracellular vesicles derived from either the normal meths or the ones induced to express OncoDibble influenced measures of transformation, including the ability to withstand serum starvation conditions, as well as down here in the bottom, soft auger growth. And so what we did is we collected the vesicles off of each of these cell types, and we put them on these normal fibroblasts. And again, here in the soft auger, as I showed you before, normal fibroblasts can't grow in soft auger. They remain as these small single cells. Interestingly, if you put on the microvesicles from the uninduced mess, so these are the normal mess, they remain as these small single cells. However, if you put on the microvesicles from the oncodibble-induced mess, this is the transformed cells, and again, they've only been transformed for a very short period of time, now these microvesicles can cause these 3T3 cells to start forming colonies. So we immediately know there's got to be something different about the microvesicles that are shed by these transformed cells compared to the normal. We found something very similar in our apoptotic assays. So in this case, again, the normal fibroblasts, if you serum starve them, they die. If we incubate them with the vesicles from the normal mess, derived from the normal mess, we can get about a 20 to 25% reduction in cell death. So they will promote cell survival to some degree. Increasing that amount, doubling that amount doesn't really change much, so we're already at a maximal effect. However, if we take the vesicles off of these induced mess, so these ones that are transformed, we get a very strong survival response. In fact, cell death rates are reduced by 80% compared to controls. So this is really telling us collectively that there's something unique, likely the cargo in these microvesicles off these transformed cells contains something that is allowing them to transform recipient cells. And so we set up screens to identify what that was. And one of the candidates that we identified was a protein known as FAK, and FAK stands for focal adhesion kinase. It's a ubiquitously expressed non-receptor tyrosine kinase. You can see here are the MVs derived from the control mess, and then here are the microvesicles from the mess from the MVs from the mess expressing oncodibble. And you can see this high enrichment of FAK in the microvesicles. And we've done this over a number of experiments, and we always see about a 12-fold increase, at least, in these vesicles. And the other really interesting thing that we found is that these vesicles, it's specific for what we see in the microvesicles, and the exosomes that we isolate from these same cells do not contain FAK. So it appears FAK appears to be only in the microvesicles from the transformed cells and only in that class of extracellular vesicle. They're lacking from the exosome derived from the same cells. And this is one of the few examples where we have found a protein that's exclusively expressed in only one type of extracellular vesicle. Now, just to give you a little bit more background about FAK, it's found in focal adhesions, or referred to as FAs, and these are sites of cell attachment and signaling hubs in virtually all cell types. And how it works is sort of depicted down here in this diagram. So there's an inactive conformation of FAK, and in response to extracellular signals, FAK can become phosphorylated at tyrosine 397. This induces a conformational change in FAK and a partial activation of FAK. This tyrosine phosphorylation then allows, this conformational change then allows another non-receptor tyrosine kinase called C-SARC to come in, form a complex with FAK, and SARC further phosphorylates FAK, leading to a fully active signaling complex. This FAK-SARC complex can then go on to activate a number of proteins found in focal adhesions. There are hundreds of proteins identified as residing in focal adhesions. One of them is paxillin, for example, and this can go on to regulate a number of cellular processes related to cancer progression. Now, what's really interesting about finding FAK in there, we also went to look to see whether SARC might also be there since it's a major FAK binding partner. And what we found is that C-SARC is expressed in the microvesicles from the transformed cells, but it's also present in the vesicles that we isolated from our control cells. So it's there all the time. And now the idea is we might just be adding FAK on top of it and could be potentially adding a signaling complex. But not all focal adhesion proteins are in these microvesicles. So for example, paxillin, which I told you FAKs are combined and activate, is not found in these microvesicles. So there is specificity certainly involved in this, and certainly not just all focal adhesions are going in at the same time. So, of course, the question came up is what role does FAK have, does this enriched FAK have in mediating the transforming effects of the vesicles isolated from the oncodibble expressing cells? And we set out to answer that by depleting FAK from the microvesicles from oncodibble expressing mess. And we can do that by transfecting these cells with a FAK siRNA You can see that in the whole cell lysates that we can deplete FAK expression by somewhere between 50 and 70%. The microvesicles that these cells then produce are also depleted of FAK. This is a reduction of about 90%. And then if we take the microvesicles off the control cells, in this case, this is the transform mess, not expressing the FAK siRNA, or after you've knocked down FAK from them, and we add them to NH3T3 fibroblasts that have been serum starved, we ask what role would the FAK play, and that's shown here. So in blue is a control again. These are cells that are just serum starved, they're dying. The microvesicles from the transformed cells promote survival, but depleting those vesicles of FAK alone is enough to lose this effect. So the FAK that's in these vesicles is clearly playing an important role in mediating their ability to promote survival and growth. But we knew it had to be more than just transferring FAK, because FAK, as I had mentioned earlier, is a ubiquitously expressed protein and is expressed in 3T3 cells at a pretty high level. And also, the amount of FAK that would be transferred via the microvesicles is not that much. So it's hard to imagine how adding just a little bit more FAK to a normal fibroblast would be enough to transform it. But then, as you remember, and I've already sort of alluded to this in the diagram, we started to wonder whether you weren't just transferring FAK, but were we transferring an activated form of FAK? And in particular, were we transferring a complex of FAK and SARC together, which is a fully active complex? Now, this would be a unique way to send signaling mechanism or a signaling protein to a recipient cell. And so what we did is we took advantage of an antibody that recognizes FAK only when it's tyrosine phosphorylated on 397, and this is important for its activation. And we probed the microvesicles from the transformed cells, and you can see here that the FAK that's in these microvesicles is indeed phosphorylated, indicating it's active. If you also take these microvesicles and we add them to a recipient cell, in this case it's normal fibroblasts, we can see that there's a higher level of FAK activity in these cells after we treat them with microvesicles from the oncodibble expressing mess, and also a downstream signaling pathway, AKT, also gets activated when we add these vesicles. And notably, the vesicles off of the control cells, which don't have FAK, are unable to induce FAK activity in recipient cells and also AKT activation. So what we're really thinking is happening now is that these microvesicles, once you, off of these transformed cells, are getting loaded with both FAK and SARC, and that what's happening is we're transferring a fully active signaling complex to recipient cells, and then that is responsible for the transforming features that they can bestow upon recipient cells. And so in order to prove that, we took advantage of a FAK2 inhibitor, in which case this is a very tightly binding FAK inhibitor. It binds very tightly to FAK and inactivates it. And so what we can do is we can take the microvesicles off of these transformed mets, we can incubate them with this inhibitor, and then we can repurify the microvesicles. So what happens is any unbound inhibitor goes away, and now all we have left is the inhibitor bound to FAK in the vesicles, and we can show that it inactivates it efficiently over here in this western blot. So these are vesicles just off of the transform mets. You can see the expression of FAK is there and also that it's active. But once we treat them with this inhibitor and then repurify them, the activation of FAK goes away, but the total amount of FAK doesn't change. So we then took advantage of these two vesicles and we added them to recipient cells in soft agar and also in a survival assay. And you can see here that the microvesicles of the transformed cells, again, promote soft agar growth of recipient cells, but if we just inhibit this FAK activity, they no longer can promote soft agar growth. And similarly, we see the same thing in the survival assay. Here's the survival ability or the survival promoting aspect of the vesicles from the transformed cells, and that's completely lost when we just inactivate the FAK that's present in these microvesicles. So that's really starting to argue that FAK is a protein that could get loaded into microvesicles very on in transformation. It's active, and it can be important for mediating the biological effects that we see with these vesicles. Now, this is interesting, but we're also starting to ask, could this be an early marker of a patient with cancer? And now we're at the very earliest stages of this, but we have done some analyses on some breast cancer cells, these three, and also on a normal mammary epithelial cell line known as MCF10As. What's interesting is that all of these cells express FAK to a similar level. And each of these cell types are generating microvesicles, as we can detect with flotilin. However, when we look at FAK in the vesicles, we see something different. So the MCF10As, these are the normal mammary epithelials, don't have FAK, express FAK, but each of the cancer cell lines do. So we're starting to wonder, could FAK be a marker of cancer in a patient? And so we're starting to set up some experiments to test that right now. So just to conclude this part of my talk, we have developed an in vitro model of cellular transformation where we can convert a normal cell into a transformed cell in a very short period of time, on the order of 1 or 2 days, and that we compared the exosomes that are generated, or sorry, the microvesicles that are generated by these cells, both the normal cells and the cancer cells that have just been transformed. The cells have been just transformed. And what we have found is that the extracellular vesicle amounts produced by each of these cell types doesn't change, but their cargo does. And in particular, we found that there is an active signaling complex of both C-SARC and FAK that can be transferred to these recipient cells and induce a transformed phenotype in normal cells around the tumor that would be localized in the tumor. And like I said earlier, now what we're really positioned to do is we can ask if FAK can be detected in MVs derived from a number of different cancer types, low-grade, high-grade tumors, and that would define whether it could be used as an early and then potentially a general indicator of cancer progression. So now, in the second part of my talk, I want to tell you a little story about that we've been working on involving this smaller class of extracellular vesicles known as exosomes, and here depicted in yellow. And we wanted to ask, what role might they be playing in cancer progression, given that we've defined microvesicles as playing an important role in cancer progression? How are the exosomes influencing this process? And in particular, what we've done is we took MDA and B231 cells. This is a highly aggressive breast cancer cell line. You take the conditioned media. We go through the exact same process where we retain the microvesicles on top of this 0.2 micron filter. But then in order to isolate the exosomes, all you have to do is take the flow through, and we subject the flow through to a centrifugation at 100,000 G for about 2 hours. And that pellets exosomes in there. And you can see that our preps are pretty pure. So these are the exosomes and microvesicles collected from the conditioned media of 231 cells. Flotilin, I had mentioned, is a marker. It's a general extracellular marker, so it's present in both the exosomes and MV preps. But when we reprobe this blot for a exosomal specific marker known as CD63, we can see that only in our exosomes, and it's lacking from our microvesicle prep. So now we can go on and study the exosomes that are produced by this highly aggressive breast cancer cell after removing the microvesicle. So we're exclusively studying the exosomes in this context. The first thing we did is we looked at the sizes of these exosomes, and they averaged a majority in between 30 and 40 nanometers in diameter. That's within the predicted sizes of exosomes. And we also did a survival assay that we did with the microvesicles. And the exosomes off these highly aggressive breast cancer cell line could promote the survival of recipient cells under serum-starved conditions, but not as well as the microvesicles did. But at least we could get some effect. But what we started to wonder is, one of the lines of study that we pursued is, how might exosome production by cancer cells change if you were to treat a cancer cell with a chemotherapeutic agent? And in particular, breast cancer cells are often, breast cancer patients are often treated with paclitaxel, also known as Taxol, and I'll refer to it as Taxol for the rest of my talk. And this is a member of the taxane class of drugs. I had mentioned it's used to treat breast cancer as well as a host of other cancer types. And its mechanism of action is the disruption of normal microtubule dynamics. And eventually what this leads to is cell cycle arrest and apoptosis. And in particular, what Taxol does is it stabilizes microtubule formation. And so I can give you a good example of this is if you take these 231 cells and you either treat them with DMSO or you treat them with Taxol and then we stain for tubulin, which will label microtubules, you could see sort of the effects of Taxol down here in these immunofluorescent images. So on the left-hand side is the control 231 cell. You see it has this polarized phenotype. It's elongated. You can see that the microtubules are sort of transversing this cell, and they're found all throughout it. However, if we treat with a really low level of Taxol, you can see that this polarized morphology of the 231 cell is lost. So these cells tend to get more rounded. They're still large, but they get more rounded. And you can certainly see all of these microtubule accumulating in these cells. I mean, there's just a tremendous accumulation of them. And that's consistent with what Taxol does as a microtubule stabilizer. So then what we wanted to know is treating cancer cells with this low level of Taxol, how would that influence the ability of these cancer cells to generate exosomes? So the first thing we did is we looked at the sizes. The sizes were about the same as the control cells, but then we quantified the numbers. And what's interesting is that after you treat with Taxol for as little as even just a couple hours, the numbers of exosomes produced by these 231 breast cancer cells is doubled. So it was quite remarkable that there's that quick of a response to this chemotherapeutic agent. And also, in addition to increasing the numbers, the function of these vesicles also changed. So I had showed you earlier that if you take the exosomes off of 231 cells that were just treated with DMSO, they will promote the survival of recipient cells that are serum starved. They'll reduce it by about 30%. If I increase that number even up from 0.5 to 1.5 times 10 to the 6th, that number doesn't change dramatically. However, if we take the vesicles off the paclitaxel treated 231 cells, they very strongly promote cell survival. So once again, this seems like Taxol treatment is causing changes in the cargo of the exosomes, in addition to actually making more as well. And so we set up a screen to ask what might be present in the exosomes from paclitaxel or Taxol-treated 231 cells. And one of the proteins that we identified in our screen is called Survivin. You could see that Survivin is highly enriched in the exosomes from the Taxol-treated 231 cells. It's often barely detectable in the exosomes from just DMSO-treated control cells. And importantly, over a number of experiments, there's about a 30-fold increase from across a number of experiments. So what is Survivin, and why did we instantly become interested in it? Well, it's a member of the inhibitor of apoptosis family. It's the IAP family. And its expression is correlated with therapy resistance and poor patient prognosis. And how it works is it resides in the mitochondria, and in response to cell death stimuli, it gets recruited to the cytosol, where it can interact with other members of the IAP family. And one of its functions is that it can directly interact with caspases and inhibit cell death. And so you can understand why it's so often upregulated in cancer cells, because it promotes cell survival by inhibiting apoptotic machinery. So two additional experiments that we did on looking at Survivin and exosomes is we asked first, is this a common outcome? If we treated cancer cells, other cancer cells, will we see a similar increase in Survivin in their exosomes? And the answer to that question is yes. So we treated a U87 glioblastoma cell line, as well as another breast cancer cell line called SKBR3 with Taxol, and we collected the exosomes, and you could see that we can detect Survivin in the exosomes only after we treated these cells with Taxol. So the enrichment of Survivin is a common outcome of treating cancer cells with Taxol. The second question that we had, which I was really surprised by these results, is the fact that we were interested in knowing whether Taxol treatment really served as sort of a general stress to the cancer cells. And so as a result, they made exosomes containing Survivin, or whether this could be a specific outcome of Taxol affecting the microtubules or stabilizing them in the cell. And so to sort of sort that out, we actually treated 231 cells with a host of other chemotherapeutic agents and inhibitors known to block the activities of proteins that are important for promoting cell growth and survival. So among these are sort of the chemotherapeutic agents, the classic ones, doxorubicin, cisplatin, and etoposide. We also have EGF receptor inhibitors, as well as HSP90 inhibitors, as well as metabolic enzymes. And all of these are known to block the growth of the 231 cells. And what we were surprised to find is that all of the cells were still capable of generating exosomes, as indicated here by the presence of flotilin. But none of these treatments could recapitulate the effects of Taxol and cause this really robust enrichment of survivin in the exosomes. So it really turns out that this is a specific outcome of Taxol treatment. Now, the one inhibitor that we tested that actually caused an enrichment of survivin in the exosomes is called nicotazole. So you can see it looks just, in the terms of amount of survivin, you can see it enriches survivin in the exosomes just as much as Taxol. And what's so interesting about that finding is nicotazole is another drug that interferes with normal microtubule dynamics. So now we have two different drugs that specifically target microtubule dynamics and inhibit it, and they both cause this enrichment of survivin in their exosomes. But there's more to it than that. Both of these drugs, although they can inhibit normal microtubule dynamics, they do so in different ways. So I've already shown you earlier that Taxol is a microtubule stabilizer, so they increase the amount of microtubules present in the cells. But Taxol is a depolymerizing agent, so it inhibits microtubule polymerization and decreases the rate at which they depolymerize. So the microtubules fall apart in these cells. Yet, so whether we build up the microtubules or we're breaking them apart or depolymerizing them, called catastrophe, in either case, we get an enrichment of survivin in the exosomes. And so this really starts to argue that maybe what Taxol is doing, it's a specific effect, but it's through changing the normal dynamics of the microtubules is enough to cause these cancer cells to release this class of extracellular vesicles, these exosomes, and enrich them with survivin. So in just the last data slide I wanted to show is we went on to see whether or not the survivin that's found in the exosomes from the paclitaxel treated 231 cells was indeed important for promoting cell survival. And in particular, we were interested in asking whether it might promote chemoresistance. So if you think about this, if you're a breast cancer patient and you're getting a treatment Taxol, you start producing exosomes that contain survivin, an important question to know in that context is whether those exosomes are traveling to nearby cells, getting taken up, and becoming resistant to the drug because of the transfer of those exosomes to the recipient cells. And so we set out to test that by using as a recipient cell line a breast cancer cell line known as SKBR3 cells, and we exposed them to Taxol. And as you can see down here in this blot, or this graph, if you expose SKBR3 cells to Taxol, they'll undergo apoptosis. But if we incubate them first with the exosomes derived from Taxol-treated 231 cells, okay, so now these are exosomes that are enriched with survivin, we can inhibit the ability of Taxol to kill these SKBR3 cells to a large degree. What's important to note about this finding is that if you use the exosomes off of control 231 cells, okay, so now these are the exosomes that don't have the survivin, and we do the same experiment, these exosomes aren't capable of promoting survival. So that would argue that maybe it's the survivin in these vesicles that's mediating this outcome, and we went on to show that by first knocking down survivin from these exosomes. So you could see it here. Here's the control siRNA. You can see the survivin in the exosomes derived from the paclitaxel-treated 231 cells. If you knock down survivin from those cells and isolate the exosomes, you can see that we could deplete the amount of survivin in those exosomes. And then if we add those, the exosomes depleted of survivin to our SKBR3 cells, they now sort of behave like to control exosomes that don't have survivin. So it's clear that the survivin that's being enriched in these exosomes is really important for promoting chemoresistance, and in particular, Taxol resistance. So just to conclude the talk, what I want to do is sort of present a snapshot of a tumor microenvironment. And this could be really a microenvironment of any type of solid tumor. In particular, this is, let's just call this a breast cancer. So you have cancer cells that would be next to other cancer cells, as well as proximal to normal cells that compose the tumor microenvironment. And what our findings would suggest is that when you start to, when you first sort of acquiring a transformed phenotype, when the cancer is initiating, the initial cancer cells might not be making more extracellular vesicles, but those vesicles have a change in their cargo. And in particular, what we've identified is a signaling complex of C-SARC and FAK, which is uniquely localized in the larger extracellular vesicles, these are the microvesicles, from the cancer cells, and that they would be being transferred not only to other cancer cells, but to normal cells, and inducing a transformed phenotype in a way that would help promote cancer progression and tumor growth. Now, at some point, a breast cancer patient would be diagnosed, and one of the regimens that you would receive is Taxol treatment. And so, in the second part of the talk, you could appreciate that now what we're starting to think happens is that as you're getting a patient's getting treated with Taxol, it's causing these cancer cells to produce more exosomes. So the numbers of exosomes produced by cancer starts to increase. And not only do the number of exosomes increase, but they're enriched with a protein that's known to be important for promoting cell survival, specifically survivin. And now what happens is these survivin-containing exosomes can travel to nearby cells and promote resistance to Taxol treatment. So in a nutshell, what happens is that we're believing now that these extracellular vesicles are playing important roles at a number of different stages of cancer progression, from the very early stages all the way to late stages in drug resistance. And then the last thing is that we're also becoming attracted to this idea that, exosomes, again, are present in the circulation of cancer patients. And if you have a patient being treated with Taxol, can we detect survivin in the exosomes from the circulation of a cancer patient? And can we use that as a potential indicator of Taxol resistance? With that, I'm just going to stop and acknowledge the people that have been instrumental in this work. So we have a core group within the Sirion lab that's known as the Extracellular Vesicle Group. There's a number of fantastic postdocs and graduate students and a number of undergraduate students that I haven't included here that have contributed to the work I've shown today. But in particular, I want to acknowledge two people. The first person is Rick Sirion. So Rick is the PI of the laboratory and instrumental in the development of every aspect of this work. And the work I talked to you about today was the thesis work of Bridget Krieger, a graduate student who's just recently left the lab and started a postdoc at AstraZeneca. And so with that, I'd like to stop, and I'd take any questions that you may have.
Mark, I have one question. You showed the interaction between FAK and SARC. Are there other binding partners of FAK?
Oh, yes. So in terms of the focal adhesions, these are forming large complexes in cells, and they're certainly capable of activating a number of signaling pathways, including ERK pathways and PI3 kinase. And so what's interesting about what we found in the microvesicles is, and then also sort of speaks to the larger sort of specificity that we see with these extracellular vesicles, is the fact that that only certain proteins, signaling proteins, are ending up in extracellular vesicles, not all of them. And so, yes, I think there are going to be a number of proteins in there that could interact. We're still looking for other ones that could be in these microvesicles. But at least we think that transferring FAK and SARC together, which is sort of this really well-established signaling sort of complex, is sufficient at this point to mediate these effects.